The Global Alliance for Genomics and Health, GA4GH, Task Execution Service, TES API is a standardized schema and API designed to describe and execute batch execution tasks in various compute environments. This API aims to provide a common way for submitting and managing tasks across different systems, including on-premise high-performance compute and high-throughput computing, HPC, HTC, systems, cloud computing platforms, and hybrid environments. The TES API has been adopted by several service providers and utilized by various workflow engines, enabling research institutions to build hybrid compute systems for studying life sciences. In the context of bioinformatics and computational biology, genomic computing involves the analysis of data through the chaining of command line programs. However, each tool used in this process is a product of extensive research in different areas of specialization, written by various teams from different institutions. These tools may be decades old or still in development, with vastly different software stacks and library dependencies. To address this complexity, several groups have developed workflow engines that describe how these tasks are chained together. Biological data is generated by numerous institutions and stored at service providers, including private institutional computing and commercial cloud providers. With the average whole genome sequencing file exceeding 200 gigabytes, downloading all data to a single storage site is impractical. The TES API solves this issue by enabling researchers to easily submit and manage tasks on a variety of compute environments, thereby facilitating efficient data analysis and research in life sciences. The TES API, developed by the Global Alliance for Genomics and Health, is a flexible and extensible API designed to address diverse computational environments, including on-premises high-performance computing and high-throughput computing systems, cloud computing platforms, and hybrid environments. This API enables the secure, responsible, and effective use of genomic data by simplifying and streamlining the execution of computational workflows, reducing costs by leveraging different compute resources. The API supports multiple workflow engines, including Galaxy, Cromwell, Nextflow, and SnakeMake, for deploying workloads on TES-fronted systems. At its core, the API relies on the task, an atomic message detailing all required task parameters, including application environment, command, and file system paths. The TES specification requires these parameters to be defined due to the lack of guarantees in the execution environment. The API also supports software containerization systems, such as Docker images, to capture entire software environments and deploy them on new computer systems. This facilitates cross-platform, cross-cloud batch execution, making it possible to use a variety of compute resources and reducing the cost of running computational workflows. The TES API is highly flexible and adaptable, designed to be deployed in various infrastructures. As an open API-defined specification for HTTP servers, it allows network access to the system and enables the generation of lightweight library bindings for new programming languages. This makes it suitable for both cloud and on-premise environments, providing a valuable tool for the genomics community and other audiences that benefit from a cross-platform, cross-cloud batch execution solution. The TES API is a standardized interface that facilitates the execution of tasks in various compute environments, allowing for seamless interaction between researchers, workflow management systems, and computational resources. It supports multiple use cases such as distributing tasks across different environments and optimizing resource allocation based on task demands. Key components include computational resources, inputs, outputs, environmental variables, and command lines that define the user process. The API allows for the development of TES-compatible clients and servers independently, making it easy to integrate into existing workflow management systems and software tools. In genomic analysis, the Task Execution Service, TES, packet exemplifies the integration of inputs, outputs, and logging. The input genome data and reference genome serve as inputs, while output processed data and log file constitute the outputs. Resource definitions and executor commands are also defined. The task is executed using a bioinformatics pipeline image, specifying commands and environment variables. The logs provide a detailed record of task execution, including start and end times, exit codes, and a unique task identifier. This demonstrates the flexibility of the TES API in executing diverse tasks, from simple commands to complex workflows, by abstracting away the compute environment. The TES API enables the creation, monitoring, control, and listing of task messages through the tasks endpoint. Notably, it provides functionalities such as task cancellation and detailed information retrieval via task identifiers. This facilitates efficient task management and oversight in genomic analysis. The TES API is a comprehensive system designed to efficiently manage and execute genomic data processing tasks. It consists of three main components: the client, server, and worker node. The client initiates task submission to the server, which allocates a worker node on an HPC, HTC, or cloud infrastructure. The worker node is responsible for transferring inputs, running user code, capturing logging, and storing outputs. Customization is enabled through adjustable task listing detail levels, which can be set to minimal, basic, or full. External file transfer protocols such as S3 buckets, FTP, and HTTP URLs are supported, with available protocols listed under the storage property when querying the service info endpoint. The API utilizes Docker images for software containerization, with plans for support of other frameworks and methods. A TES task can comprise multiple command lines defined in an executor, specifying the command line, container image, working directory, stdin, stderr mapping, and environmental variables. These executors are run sequentially, and if the ignore_error flag is not set, a non-zero exit code from one executor will terminate the sequence early. This mechanism enables optimization of workflow execution by scheduling linear subworkflows on the same TES instance, allowing for efficient genomic data processing. The Task Execution Service (TES) API provides a standardized interface for task execution, facilitating the integration of diverse computational platforms and infrastructure. This enables workflow management systems like Cromwell, CWL, and Nextflow to execute tasks on various execution environments. The TES ecosystem comprises multiple server implementations, including Funnel, Pulsar, TES Azure, and Tesk, which cater to different environments such as high-performance computing, HPC, and high-throughput systems, HTS, Galaxy, Pulsar, Microsoft Azure, and Kubernetes, native cloud systems. The proxy service, ProTES, allows for injecting middleware into GA4GH TES requests, enhancing the API's functionality. Clients in the TES ecosystem, including Cromwell, CWL, Nextflow, Elixir Cloud Components, Pytes, SnakeMake, and Toil, interact with TES services and other GA4GH APIs, contributing to the development of a robust and flexible infrastructure for computational workflows. The Elixir Cloud and AAI GA4GH driver project and the European Genomic Data Infrastructure are leveraging the TES API to create a cloud infrastructure that federates the execution of computational workflows across different national nodes in the Elixir community. Microsoft has also utilized TES to support the Cromwell workflow engine on Azure, eventually splitting the implementation into its own standalone repository and deployment. The TES API is designed to be lightweight and maintainable, allowing multiple implementers to build compliant clients and servers easily. Future versions of the API aim to provide better guidance on authentication, security, and software portability. The evolution of the API will primarily involve refinements to the conformance test suite rather than changes to the OpenAPI description. By promoting interoperability among different solutions, particularly in multi-tenant environments, the TES API plays a crucial role in advancing the development of computational workflows. The TES API is a versatile, cloud-agnostic, and container-agnostic API designed to facilitate the execution of computational workflows in a hybrid environment encompassing both cloud and high-performance computing (HPC) systems. It integrates various computational tools and platforms, allowing researchers to develop their analysis workflows independently of the infrastructure and deploy them in a multi-cloud approach, targeting computational resources that are more affordable or closer to the data that needs to be processed. The API has been successfully integrated into systems such as Funnel, ProTES, and TES in Azure, demonstrating its utility in powering computational analysis for life sciences. It supports different containerization systems including Docker, LXC, Singularity, RKT, and Podman, as well as software management systems like Conda, Bioconductor, and Galaxy Toolshed. Possible extensions to the API include enabling privacy-preserving federated learning, supporting Hadoop or Spark jobs, and adding a callback API. These extensions could be added as optional extensions that would not be required for basic conformance. The TES API is designed to be interoperable, making it easy to integrate into existing cloud infrastructures, workflow management systems, and other relevant software solutions. This allows researchers to develop their analysis workflows independently of their infrastructure and deploy them in a multi-cloud approach, targeting computational resources that are more affordable or closer to the data that needs to be processed. This work was funded by grants from the National Institute of Health, including 1U24CA231877 and 1U24HG010263. The work was further supported by Elixir, the research infrastructure for life science data. Additionally, this work was supported by Microsoft's biomedical platforms and genomics team. Working group leadership includes Anya Niewielska, until 2022, K, AK, and VM. Additional API contributors include Sushil Varma, Adam Struck, Alexander Buchanan, Angel Pizarro, Gina Lee, Ryan Spangler, Jeff Gentry, Brian O'Connor, and Benedict Patton. VM and K are CO champions of the GA4GH TES API specification. Each of the authors have contributed significantly, leading role in planning, implementation and/or maintenance, to one or more of the TES ecosystem projects described in this manuscript. The Global Alliance for Genomics and Health, GA4GH, Task Execution Service, TES, API is a standardized interface designed to facilitate the execution of tasks on distributed compute resources. This API has been successfully implemented in several projects, including Microsoft's implementation, available at 6, the Oregon Health and Science University's Funnel project, accessible at 7, and the Elixir Cloud AAI-TES kit, found at 8. These implementations showcase the versatility and adaptability of the TES API, demonstrating its potential to enhance collaboration and efficiency in genomic research. The TES API operates as a middleware layer, connecting users with various compute resources across different institutions. This allows researchers to submit tasks to be executed on available computing platforms, thereby optimizing resource utilization and minimizing operational costs. By standardizing the interface for task submission and management, the TES API simplifies the process of scaling up or down according to research needs, making it an essential tool for large-scale genomic studies. Moreover, the TES API promotes transparency and reproducibility in research by providing a clear and consistent way to describe, submit, and track computational tasks. This facilitates collaboration among researchers from different institutions, enabling them to share knowledge, resources, and best practices more effectively. Furthermore, the TES API supports a wide range of computational workflows, making it suitable for various types of genomic analyses. In conclusion, the GA4GH TES API is a powerful tool for enhancing the efficiency and collaboration in genomic research. Its successful implementation in various projects demonstrates its potential to revolutionize the way researchers interact with distributed compute resources, ultimately accelerating the pace of scientific discovery in the field of genomics.